All right, so welcome to the deep dive. Today, we're going to do like a super fast tour of cells. You gave us a ton of research about prokaryotic and eukaryotic cells, and we're going to really try to like distill it down
Okay.
so that everybody listening can like walk away and be like, wow, I actually kind of get cells now.
Awesome.
And I think a lot of it was really focused on how scientists discovered cells.
Right, yeah. This is a super interesting journey, too. It starts like way back
Okay.
when people actually used to think that like life could just appear out of nowhere.
Oh, wow. So like if you find a cake on the counter,
Yeah,
you just think it like poofed into existence.
exactly. Like imagine you see, maggots on meat or something.
Right.
And you just assume, well, they must just kind of sprung up from the meat itself.
Right.
Like that was the theory of spontaneous generation.
Wow. Okay.
It seems wild now, but like back
Yeah.
then, that was
Yeah.
like the main belief.
So who was the person that like finally busted that myth?
Well, there were a few key people, but one of the first was Francesco Redi back in the 17th century.
Okay.
And he did this really simple experiment with meat and jars.
Okay.
And basically he showed that maggots only appeared when flies could actually lay their eggs on the meat.
Okay.
So it was like a really simple experiment, but it started to like poke some holes
Yeah.
in that spontaneous generation idea.
Okay. So like one experiment isn't enough to overturn centuries of belief.
No, totally. It was a gradual process. I mean, there were these heated debates
Yeah.
and tons of like conflicting results. Like there was John Needham, and he thought he'd proven spontaneous generation because he saw these microbes growing in broth that was like sealed up.
Right.
But then this other guy, Lazzaro Spallanzani, he comes along and he repeats the experiment,
Okay.
but he's extra careful, right? Like he boils the broth for longer and seals the flasks like really well.
Okay.
And guess what? No microbes.
Oh, wow.
So like they're going back and forth, each convinced that they're right. It really shows you how like
Yeah.
even a simple experiment can have all these like subtle factors that affect the outcome.
Totally. So who finally like settled it? It sounds like a real nail biter.
Well, so Louis Pasteur comes along in the 19th century and his swan neck flask experiment.
Okay.
That was like the final blow.
Mhm.
Basically, he uses these flasks with long, curved necks, and they're open to the air.
Okay.
But the microbes can't just like magically appear in the broth.
Okay.
Only when he like tipped the flask and let the broth touch the dust and microbes in that neck, that's when they started growing.
Oh, okay. So no spontaneous generation.
Yeah.
Life has to come from life. That's pretty profound.
Right.
Okay. So that like paved the way for cell theory, right?
Exactly. So
That all living things are made of cells, and cells can only come from other cells.
Right.
But wait, I thought somebody was looking at cells before Custer.
You're thinking of Robert Hooke.
Yeah.
Way back in 1660,
Yeah.
he was using this like really basic microscope.
Okay.
And he was looking at a piece of cork. Yeah. And he saw all these tiny compartments, and he called them cells because they reminded him of like little rooms that monks lived in.
Oh, like monastery cells.
Exactly.
Okay. So if he was looking at cells in the 1600s, why did it take like 200 years for cell theory to become like a thing?
Well, imagine trying to put together this huge puzzle.
Okay.
But you only have a few pieces, and your vision's kind of blurry. Microscopes back then weren't that great.
Yeah.
And scientists were only just starting to understand how complex life really is.
Yeah.
It took way more powerful microscopes and like a bunch of different scientists working together to finally, nail down cell theory.
So who were some of those scientists? Like who were the all-stars who put this puzzle together?
We've got a few. So Schleiden was a botanist, and he was studying plants.
Okay.
And he was like, hey, all plants are made of cells.
Okay.
And then Schwann, who was a zoologist studying animals, he had the same realization.
Okay.
Like they both had this aha moment where they're like, wait a minute, cells are like the basic building blocks for everything.
So that was like the first part of cell theory. Everything's made of cells. But what about the part where cells only come from other cells?
Right. So that's where Remak and Virchow come in.
Okay.
Now, Remak, a lot of people don't really know about him, but he was actually the first to provide real evidence that cells divide to create new cells, not just appear out of nowhere.
Okay.
And then you've got Virchow, who's kind of a big personality, and he loved to like sum up complex ideas in these catchy phrases. He's the one who said, omnis cellulae cellula, all cells come from cells.
Okay. That's catchy.
Right.
Yeah.
Like it really stuck.
Okay. So it was a real team effort.
Really.
Now, before we jump into the different types of cells, can we talk about the endosymbiotic theory?
Oh, of course.
Because that's one of my favorites. It's like this cellular love story, right? That shaped how life evolved.
It's a super cool concept. So imagine billions of years ago, you've got these single-celled organisms just swimming around, and then boom, one engulfs another. But instead of like digesting it completely, they form this like partnership.
So instead of being lunch, the engulfed cell became a roommate.
Yeah, exactly. Like they started sharing resources and abilities. And that's actually how scientists think mitochondria, the powerhouses of our cells, and chloroplasts.
Okay.
You know, the things that let plants do photosynthesis.
Right.
Came to be.
Oh, wow.
They were once free-living bacteria that got absorbed by other cells and eventually became these essential organelles.
That's wild. It's like a total game changer for evolution.
Oh, sure.
Okay, so we've got that foundation of cell theory and this crazy concept of endosymbiosis.
Right.
Now let's talk about prokaryotic versus eukaryotic cells. I always think about it like comparing a tiny studio apartment to a giant mansion.
Perfect analogy. So prokaryotic cells, like bacteria, they're the studio apartments.
Yeah.
Simple, compact.
Okay.
No true nucleus.
Okay.
Their DNA just hangs out in this central area called the nucleoid, like having all your stuff in one open space.
Okay, so no separate bedroom for the DNA.
Exactly.
But what about other stuff? Like, do they have a kitchen or bathroom?
Well, not in the way we think of them, but they do have a cell wall, which is kind of like, you know, their sturdy outer walls.
It's like protection.
Yeah, essential for maintaining their shape, protection. And some of them have these extra loops of DNA called plasmids.
Okay.
Which are like bonus features, you know, kind of like upgrades that can give them special abilities.
Okay, so like an add-on package on a car.
Totally. So they might be small and simple, but they've got some tricks up their sleeve.
I've also heard about endospores. Those are like time travelers, right? Some bacteria can basically hibernate when times get tough.
Oh, yeah. Endospores are these incredibly tough, like escape pods that allow some bacteria to survive crazy conditions, heat, radiation, whatever.
Wow.
They can lie dormant for years, even centuries.
Oh.
And then when conditions improve, boom, back to life.
So they're prepared for anything.
Yeah, amazing survival strategy.
Okay, let's shift to the mansions, the eukaryotic cells.
Okay.
The complex cells that make up animals, plants, fungi, and protists.
Yep.
They've got that dedicated room for their DNA, the nucleus.
You got it. Eukaryotic cells are all about organization, compartmentalization. They've got this whole intricate network of membrane-bound organelles, each with its own specific job, like a bustling city with all these different departments.
So instead of one big open space, they have rooms for everything.
Exactly.
So what kind of rooms are we talking about?
Well, you've got the endomembrane system, which is like the cellular postal service, moving molecules between different organelles. The Golgi apparatus, that's like the packaging and shipping department.
Oh, cool.
Where proteins get modified and sorted for delivery.
Okay, so it sounds like a well-oiled machine. What other departments are in this cellular city?
We've got lysosomes, which are like the recycling centers, breaking down waste and recycling molecules. Peroxisomes are like the detox units, neutralizing harmful stuff.
Oh.
And of course, we can't forget the ribosomes, the protein-making factories.
Right. Ribosomes, they're in both prokaryotic and eukaryotic cells, right?
Yeah, they are.
But are they different?
Yeah, actually, that's a good question. So both types of cells need ribosomes to make proteins.
Right.
But eukaryotic ribosomes are bigger, more complex than the prokaryotic ones.
Okay.
It's one of those subtle but important differences that shows you how much more complex eukaryotic cells are.
Okay, so we've got the basic blueprints for both cell types.
Yeah.
But what about their walls? Are there differences there too?
Yeah, definitely. They both offer protection, but they're not identical.
Okay.
With bacteria, you can actually tell different types apart based on their cell walls.
Okay.
So like gram-positive bacteria have this thick layer of peptidoglycan. Think of it like this strong mesh-like structure.
Okay, so a sturdy wall.
Yeah, exactly.
But what about gram-negative bacteria?
They've got a thinner layer of peptidoglycan, and it's sandwiched between two membranes. And this is actually super important because it affects how they respond to antibiotics.
Wait, so the structure of their walls affects how well antibiotics work?
Totally. The outer membrane of gram-negative bacteria can act as a barrier, making it harder for some antibiotics to get through and do their job.
Oh, wow.
So infections with gram-negative bacteria can be trickier to treat.
Oh, I see.
And understanding these differences is like super important for figuring out the best treatment strategies.
Wow. So even something as simple as a cell wall can have this huge impact on our health. But bacteria aren't just sitting around, are they?
Nope, they're not.
Like, I know they've got these cool appendages called flagella and teli
Right.
to move around and interact with their surroundings. Like, they've got their own tiny transportation system.
Totally. So flagella are like tiny whips that help bacteria swim through liquids.
Uh-huh.
And get this, they can even sense chemical gradients and move towards attractants. It's called chemotaxis.
Chemotaxis.
Yeah, basically it's their GPS system.
Wow. That's pretty amazing for a single-celled organism.
Right.
And what about pili?
Kepliers are more like grappling hooks
Okay.
that help bacteria attach to surfaces. They're also involved in exchanging genetic material between bacteria, which is, how antibiotic resistance can spread so quickly.
So it's like a microscopic handshake that can have some major consequences. Okay, so we've covered their defenses, their transportation, and even how they share information.
Right.
Now let's go inside those eukaryotic mansions, right?
Yeah.
What other cool structure are hiding in there?
Oh, there's so much more to explore.
All right. So we'll dive into all of that next time on The Deep Dive.
Sounds good.
All right. So welcome back to The Deep Dive. Last time, we were exploring this incredible world of cells, right?
Yeah, we covered a lot of ground.
We did. We debunked spontaneous generation.
Right.
We talked about cell theory.
Huh.
We even compared those, simple prokaryotic cells
Yeah.
to those big, complex eukaryotic cells.
Like studio apartments versus mansions.
Exactly.
Oh.
And we even touched on endosymbiosis.
Right. One of my favorite topics.
I know. It's so cool. It's like these ancient cells.
Sure.
They knew how to team up to create something amazing.
Totally.
So now we're ready to go deeper into those eukaryotic mansions, right?
Okay, let's do it.
We're talking organelles, those specialized compartments that make eukaryotic cells so efficient.
Right. It's all about division of labor. Like each organelle has its own specific job to do.
Okay,
Just like departments in a company.
got it. So where do we start?
Well, I think it makes sense to start with the control center, the nucleus.
Ah, the nucleus. That's where the DNA is, right? Like the CEO's office.
Exactly. It's this membrane-bound compartment
Okay.
that holds all the cell's DNA, those precious blueprints of life.
Okay. But it's not just storage, right?
Oh, no, it's much more dynamic than that.
Okay.
Think of it like a library where information's constantly being accessed, transcribed, put to work.
Okay, so we've got those blueprints in the nucleus, but how do they actually get used to build proteins?
Ah, well, that's where protein synthesis comes in.
Yeah.
This amazing two-step process involving transcription and translation.
Okay, break that down for me.
So first, the DNA code gets transcribed into a messenger molecule called RNA.
Okay.
Think of RNA like a copy of the recipe, but written in a language the ribosomes can understand.
Okay, so the RNA carries those instructions from the nucleus out to the ribosomes.
Exactly.
Those protein-making factories we talked about.
Okay.
Okay, and then what?
So the ribosomes, they read that RNA message,
Uh-huh.
and using the cell's supply of amino acids as building blocks, they assemble the protein.
Yeah.
Based on those instructions, kind of like a complex assembly line.
Oh, that's really cool. So all this activity must take a ton of energy.
You're telling me cells are like bustling cities. They need constant fuel.
So where do they get that fuel?
Well, remember those ancient bacteria that became the powerhouses of the cell?
The mitochondria.
You got it. That's where cellular respiration happens.
Okay, remind me, how does that work again?
Well, basically glucose, a simple sugar, gets broken down. And this releases energy in the form of ATP.
ATP.
Yeah, it's like the cell's energy currency, those little packets of fuel that power everything.
Oh, wow. So mitochondria are like tiny power plants, converting fuel into usable energy.
Exactly. Pretty amazing, right?
Yeah.
A process that evolved billions of years ago.
Yeah.
Still powering every breath we take, every muscle we move, every thought we think.
That's incredible. Okay, but what about those other organelles? We've got the endomembrane system, Golgi apparatus.
Right.
Lysosomes, peroxisomes. It's a whole bustling city in there.
It really is. So the endomembrane system,
Okay.
it acts like a network of highways and transportation systems,
Okay.
connecting different organelles and making sure molecules get to where they need to be.
Okay, so like a delivery service.
Yeah. And then you've got the Golgi apparatus,
Mhm.
which is like a packaging and distribution center, modifying and sorting proteins.
So it's all super organized.
Oh, for sure. Everything has its place and purpose.
That's amazing. And those cellular recycling centers and detox units.
Right. Lysosomes are like the waste disposal system, breaking down cellular debris.
Okay.
And peroxisomes are like specialized detox units. They neutralize harmful substances, protect the cell from damage.
So it's like a perfectly choreographed symphony, right? All these organelles working together.
It really is.
But how do they communicate? I mean, they can't just shout across the cytoplasm.
No, they've got much more sophisticated ways of communicating.
Okay, so how do they do it?
Well, they use a combination of chemical signals,
Okay.
physical contact, even electrical impulses.
Wow. It's like they've got their own internal messaging system.
Right. They're constantly sending signals, sharing information, coordinating activities.
Okay, so we've talked about how organelles talk to each other. But what about cells communicating with other cells?
Oh, yeah, that's crucial too, especially in multicellular organisms like us.
Right. So how do they do that?
Well, there are a few different ways. Some cells communicate through direct contact, like shaking hands. Others release signaling molecules, kind of like messages in a bottle,
Okay.
that travel to other cells and trigger specific responses. And some cells even use electrical impulses.
So like a cellular text message?
Exactly. Fast and efficient communication over long distances.
It's amazing. They've got this whole repertoire of communication strategies. Okay, but we can't forget about one of the most fundamental processes, cell division. How do cells make more of themselves?
Right. That's essential for growth, development, repair, all the things. When a cell divides, it creates two identical daughter cells,
Okay.
each with a complete set of DNA.
That's like a perfectly executed dance routine, right? Everything has to happen in the right order.
Totally. But there are actually two types of cell division, mitosis and meiosis.
Oh, right. I remember reading about that. What's the difference again?
So mitosis is the type of cell division used for growth and repair.
Okay.
It creates two identical daughter cells from a single parent cell.
Okay, so mitosis is all about making copies.
Exactly. It's happening all the time in our bodies, replacing old cells, helping tissues grow and heal.
Cool. What about meiosis?
Meiosis is a bit more specialized. It's only involved in sexual reproduction.
Okay.
It creates gametes, sperm, and egg cells, which only have half the number of chromosomes as other cells.
Wait, why half?
Well, it's all about ensuring that when a sperm and egg cell combine,
Yeah.
the resulting fertilized egg has the correct number of chromosomes.
Oh, I see. So if each gamete had a full set, the zygote would have double.
Exactly. And that wouldn't be good.
No, that wouldn't work out well. So meiosis is like this cellular matchmaking process, making sure the next generation gets the right amount of genetic information.
Exactly. It's all about balance and keeping things running smoothly.
Wow. So from DNA replication to protein synthesis, energy production, waste management, communication, and cell division, it's clear that cells are incredibly dynamic and sophisticated.
For sure. They're like these tiny universes constantly buzzing with activity.
But this knowledge isn't just about appreciating the microscopic wonders of life.
No, definitely not.
It also has some really important real-world applications.
Absolutely.
Ready to explore how our understanding of cells is shaping the future of medicine, agriculture, and even energy production.
Let's do it.
Welcome back to the Deep Dive. So we've been exploring this amazing world of cells.
Yeah, from their ancient origins to their, crazy complex inner workings.
Right. But now it's time to see how all this microscopic knowledge is actually, impacting the real world.
Yeah. It's really remarkable how understanding these tiny units of life can lead to, huge breakthroughs in so many different fields, like medicine, agriculture, energy, even environmental conservation.
Okay, well, let's start with medicine, because I feel like that's where most people see that connection.
Yeah.
If you understand how cells work,
Right.
then you can understand how diseases develop,
Right.
and then how to fight them.
Exactly. Take antibiotics, for example. They work by targeting very specific bacterial structures or processes, like cell wall synthesis or protein production,
Yeah.
to basically stop those bacteria from growing.
Okay.
And the thing is, because we understand these cellular mechanisms, we can constantly develop new, more effective antibiotics
Okay.
to combat that growing threat of antibiotic resistance.
So it's like a constant, back and forth.
Yeah, kind of like a microscopic arms race.
Right. But bacteria aren't the only bad guys out there, right? What about viruses?
Oh, yeah. Viruses are tricky.
They're not even cells,
Right.
but they still hijack our cells
They do.
to, replicate and turn them into little virus factories?
Exactly. So viruses are these little packets of genetic material that need a host cell,
Okay.
to survive and reproduce. That's why antiviral drugs
Yeah.
are designed to interfere with specific steps in that viral replication cycle.
Okay.
It's like throwing a wrench in the machinery.
Got it. So cell biology is like essential for figuring out how to target those viruses. Okay, so we've talked about infectious diseases.
Right.
But what about things like cancer?
Yeah, cancer is a whole other challenge.
It is.
Because it's a disease of uncontrolled cell growth.
Right.
But our understanding of the cell cycle, how cells regulate their growth and all the signaling pathways involved,
Okay.
that's leading to some amazing breakthroughs in cancer treatment.
Okay, like what?
Well, like targeted therapies, for example.
Yeah.
These therapies can actually zero in on cancer cells specifically,
Okay.
sparing the healthy cells, which means fewer of those awful side effects
Oh, wow. Okay.
you often see with traditional chemotherapy.
That's amazing. And I've heard about immunotherapy, which is like basically boosting your own immune system to fight cancer.
Right. Immunotherapy is incredibly exciting because it uses the power of the body's own defenses
Yeah.
to recognize and destroy cancer cells.
Okay, so how does that actually work?
Well, by understanding how immune cells interact with cancer cells,
Yeah.
we can develop therapies that, boost the immune system's natural ability to fight the tumor.
So it's like training a special forces team of cells.
Exactly. To go after those cancer cells.
That's amazing. Okay, so we see how cell biology is like at the heart of all these advances in personalized medicine.
Absolutely.
But let's move beyond medicine for a second. What about agriculture?
Oh, yeah, there are tons of applications there, too.
Like, I've heard about genetically modified crops
Right.
that are more resistant to pests and diseases.
Yeah, that's a great example. By manipulating genes that control things like plant growth,
Okay.
development, and defense mechanisms, scientists can create crops that are more nutritious,
Okay.
produce higher yields, and are just better equipped to handle environmental stress.
So things like drought or pests.
Exactly.
So we could have crops that thrive in really harsh conditions.
Yeah, it's a game changer for global food security. And it doesn't stop there.
Okay.
Scientists are even exploring ways to engineer algae and other microorganisms to produce biofuels.
Oh, wow.
So we could actually replace fossil fuels.
So it's like cell biology is leading us towards a more sustainable future.
Right. I mean, imagine a world powered by tiny cells.
That's amazing. But it's not just about using cells for human benefit, right?
No, you're right.
We also need to understand and protect that incredible diversity of life on Earth.
Absolutely. By studying cells and the molecular mechanisms that drive biodiversity,
Yeah.
we can unlock the secrets of evolution, learn how different species are related,
Okay.
and then develop strategies to conserve endangered species
Right.
and those fragile ecosystems.
So from medicine and agriculture to energy and conservation, it's clear that cell biology is the key to solving some of the world's biggest challenges.
Yeah, it's pretty amazing to think that something so small can have such a huge impact
Right. It really is.
on our lives and the future of our planet.
So we started this deep dive just wanting to understand the basics of cells.
Right.
And now we've gone on this journey through history. Yeah. Evolution and the future of scientific innovation.
It's been quite a ride.
It really has. We've seen how understanding cell biology has revolutionized medicine, it's transforming agriculture, and it holds the key to a more sustainable future.
It does.
But I think most importantly, what we want you to take away from this is just a sense of awe
Right.
for these tiny building blocks of life.
Because within each cell, a whole universe unfolds, this incredible symphony of molecular interactions that makes life possible.
And as we keep exploring this microscopic world, who knows what other amazing discoveries await. So keep those minds curious and those imaginations ignited. We'll see you next time on The Deep Dive.